Hi everyone. In today's video, we will be sharing with you the 5 common mistakes to avoid during PCR. Mistake number 1 is contamination. Always remember to keep your work area clean and sterile. Use separate pipette and tips for each sample to prevent cross-contamination. The mistake number 2 is incorrect primer concentration. Ensure that your primer are properly diluted to recommended concentration. Too much or too little can affect the efficiency of your PCR reaction. Mistake number 3 is the use of insufficient template of DNA. Make sure to add the right amount of template DNA to your PCR reaction. Too little can lead to the weak or no amplification, while too much can cause non-specific amplification. The mistake number 4 is inadequate denaturation. It's crucial to ensure proper denaturation of your DNA template. Follow the recommended denaturation temperature and the time for your specific PCR protocol. And lastly, mistake number 5 is incorrect annealing temperature. Use the appropriate annealing temperature for your primers. Too high or too low can result in the non-specific amplification or no amplification at all. Remember, by avoiding these 5 common mistakes, you will increase your chances of achieving accurate and reliable PCR result. Thank you for watching. If you found this video helpful, don't forget to like and subscribe the channel and hit the bell icon for the more scientific tips.